Hello. In this video lecture, we will talk about bacterial capsules. So let's first talk about water capsules, slime layers, and glycocalyx. These are all related terms. Capsules are extracellular gelatinous polysaccharide-like materials secreted by some bacteria outside the cell envelope. They are either tightly or loosely adhered to the cell wall, and accordingly they are known as capsules or slime layers. If they are tightly adhered, they will be called capsules, and loosely bound, which can easily part off from the cell, can be peeled off from the cell, then it will be called slime layers. Slime layers are also called extracellular polysaccharides or exopolysaccharides, EPSs. So if this is a bacterial cell, then this is the cell wall around the bacteria, and the capsules are present outside that cell wall. So this is the capsule, this structure, and if the capsules can be of two types depending on the thickness. If the capsule is very much thicker, if it is more than 20 nanometer rather thick, then it will be called macrocapsule, and if it is less than zero point 0.2 micrometer, means 20 nanometer, it is called microcapsule. In fact, slime layers are much thicker than the capsule, but they are loosely bound and they can be erased. They are not covalently linked with the cell wall, whereas the capsules are covalently tagged with the cell wall material, the N-acetyl glucosamine and the N-acetyl muramic acid residues of the cell wall, that is the peptidoglycan. So capsules are usually formed by the bacteria that form a mucoid growth in the agar plates. So here is the picture showing mucoid growth of bacterial colonies of Klebsiella that also produces a copious amount of capsules. So because the capsules are made of polysaccharide, that is carbohydrate, and they are spell code one type of, that's why they are also called glycocalyx. How can you visualize capsules? A particular strain of cell contains capsule or not? Staining procedure are there for that. If you stain the cell, it is called negative staining. If you stain the cell with stains like India ink or Negrosine or Congo red, the stain doesn't penetrate the capsule. It remains outside in the background as well as it enters into the cell also. So the capsule region remains, it becomes unstained and it is visible in the dark background. This is called negative staining. In this way you can visualize the capsules and you can distinguish between a capsular and non-capsular bacteria. This can be a taxonomic character also. So now let us see the chemical composition of capsules. Capsules are usually made of polysaccharides, and two types of polysaccharides are there. The homopolysaccharides, that they are made of similar types of monomers or subunits. The heteropolysaccharides, they are made of different types of subunits or monomers. So what different kinds of homopolysaccharides are found? Let us see some examples like the dextrans, levans, cardlan. These are the different kinds of homopolysaccharides found in different types of bacteria in their capsules. Dextrans are made of glucose. They are branched molecules, macromolecules made of glucose. The main chain is alpha 1,6 glycosidic length, and the branches are alpha 1,3 with 1,3 linkage. They can be observed in bacteria like Pediococcus, Leuconostoc mesenteroides, etc. Levans are made of polyfructoses. They can be visualized. There are alpha 2,6 linkages present in the fructoses, and they make the levans. They can be seen in Pseudomonas species. Cardlan are made of glucose, and they can be seen in Rhizobium meliloti and and Agrobacterium radiobacter, this particular species. So these are different types of homopolysaccharides that you can find in different types of bacteria in the capsules. Now some heteropolysaccharides, uh, example of them is xanthan, which is made of glucose, mannose, and glucuronic acid in different ratios. It can be found in the capsules of Xanthomonas. Uh, Succinoglycan, which is made up of glucose and galactose. It can be found in the capsules of Alkaligenes faecalis. And alginic acid or alginate, made up of glucuronic acid and mannuronic acid, is a composition of Pseudomonas aeruginosa capsules. Exceptionally, there are some bacteria, for example, the Bacillus anthracis, the causal organism of anthrax disease. It contains a capsule which is made of polypeptide. It is not a polysaccharide, it is polypeptide and made of poly-L-glutamic acid residues. So this particular bacterium, this is an exception. Otherwise, normally the capsules are made of polysaccharides. Now, let's see the functions of capsules. There are bizarre functions of capsules. Number one, capsule protects cells against dehydration or desiccation because capsule is a material which can absorb 90%, 95% water of its volume, and that's why it protects the cell from getting dry. It also protects the cell from losing its nutrients into the environment. Um, Capsules provide protection against injuries, and also phages, bacteriophages cannot attach because cannot penetrate into the cell because of the capsule in many cases. However, there are bacteriophages that can dissolve the capsule and can enter into the phage, dissolving the cell wall material and the membrane also. So that is a different thing, but in many cases it protects from bacteriophage attack. Capsules increase virulence in our body of pathogenic bacteria. They evade, the capsules help the bacteria to evade phagocytosis. The first round of immune response that our body develops against the pathogens is the innate response, among which the phagocytosis is one of the most important things, which is mediated by the cells like macrophages or neutrophils, dendritic cells, etc. They phagocytose the incoming pathogens, and then they digest them with the help of the lysosome, or they process the antigenic part, the epitope part of the antigen, and then present them with the help of the major histocompatibility complex receptors (MHCs). So capsular bacteria, they are more pathogenic, they are virulent compared to their non-capsular counterparts. Due to the presence of the capsule, lysozyme enzyme that breaks down the beta 1,3 glycosidic bond between the naganama subunits and the peptidoglycan, the capsule protects the cell from getting degraded due to lysozyme activity. Capsule acts as an antigen, it's called K antigen. It triggers antibody response in the host. Capsules have to adhere on the smooth surfaces in the natural environment, and because of it helps in the formation of biofilms, which are very crucial for survival of the organisms, microorganisms, particularly in an environment which is not friendly. So these biofilms, they are colonies of bacteria, and they are formed within a gelatinous material. There is the exopolysaccharide or the capsule that is released by the bacteria, and it forms a coating around the biofilm, because of which the harsh chemicals, even the antibiotics, cannot penetrate it, and the bacteria, the pathogen or the bacteria, it shows antibiotic resistance because of this also. Uh, the dental carie is caused by this Streptococcus mutans. Is because of this reason. These bacteria, Streptococcus mutans, they secrete a capsule which is the homopolysaccharide made of mutants. Mutants are also one type of polysaccharides made of glucose. So This causes the bacteria to colonize that particular place and cause pathogenesis. Lastly, sometimes bacteria use capsules as source of nutrients when there is lack of energy in the cell. Then the cell can utilize this as a source of nutrient, can excrete, it can secrete enzymes to dissolve them and can absorb the nutrients. The monomers released by the degradation of polymers, polysaccharides, like this S mutants, may use its capsule as a source of nutrition by breaking it down and utilizing the sugars when energy stores are low. So these are the different functions of capsules, and that's all about capsule. Thank you very much. Hope you like this video lecture, and I hope you will subscribe the channel and also click the bell button to get more notifications about similar video lectures in future. Thank you very much.